Yeah, whatever.
Good evening, everybody. We're at SNATA right now, and we have Speed, our beautiful host. We have Nina guy, Scott, always a pleasure, and we have Lauren or Lori, which
Yeah, we know how he is, right? Yeah.
What just happened to your voice, Midnight? You just went weak on us. What the fuck?
Oh, I lowered his mic setting because it was going way over the top.
Oh, you lowered it all.
Yeah, so there we go.
Okay. Yeah, sorry, Scott.
Oh, no problem. No problem. I'm just, adjusting a little.
Can I finish?
No.
Can I finish?
No.
God damn it.
I hate all of you. Just kidding.
I hate everybody today myself. Pretty
Why?
mad.
We love you.
I don't know. It's 85 degrees, fucking windy. We're going to, I don't know what's happening out there.
Wait, we just started. We're going to shoot it in the head already?
I think we should shoot it in the head. I don't know. We should quote while we're ahead. So, Gerstream last night went 10 and a half hours, and it was...
yes, it was insane.
There was a lot of interesting stuff thrown out there. The thumbnail is from a paper that John Maddox has been going nuts about, this 13-bit Talin code, which doesn't make a whole lot of sense with the way they're portraying it. But as it turns out, the idea and what's happening does make a lot of sense applied to other parts of neuroscience. So anyway, it's a paper to watch. It's a good paper. It's
you mean read?
Well, no, you just got to watch this guy and his theory. I think his name is, I forget what the fuck his name is, but I could figure it out here if I pull the
Penrose?
paper up. No, it goes to show, for the same reason that he's kind of wrong about what he's saying, Penrose is wrong for the very same reason. And it's sort of hard to figure out.
Andrew, you are proof of God via Tangelos as Holio. Yeah, I'm gonna say Peter.
Okay, this guy's name is Goult. Goult. He's a young researcher, and he works on basically focal adhesions in the cell and cell skeletons, cell structures, and there's something called actin, which is probably the scaffolding of your cell. And there are a bunch of other proteins that hold that actin together. And one of them is called Talin. And it has 13 different switches in it that increase the amount of pressure that it exerts on the proteins that it attaches to. So it's kind of like a little spring. But it's also a spring with memory. So once you extend it to a certain spot, apparently it's thermodynamically stable and it stays there. And it's very, very interesting. So anyway.
Well, hold on. What does that mean to be thermodynamically stable?
That means that it'll stay in that state. The state doesn't degrade. So if 9 out of 13 of these domains are in the folded state, then it will keep that. It'll remember that. So it provides a little bit of memory for neurons, which is interesting because one of the things neurons do is if a neuron fires, well, if you see a rabbit and then you look away and see something that's similar to a rabbit, your rabbit neuron is very likely to fire again. Okay. It's basically post-tectonic, tectonic So potentiation is what it's called. So once you've seen something in your brain keeps a little short memory of things that are currently going on. And they've had a lot, I mean, they've been working for years and years to detail all of the mechanisms that do that. And then the other thing that happens is if that neuron fires at a certain frequency over and over again, it can actually change your synapse. So for the rest of your life, there will be a change to that particular part of your brain. So, and that's kind of how memory works. So it works at the synapse. So he has a lot of information on that. But Maddox is like, it's just another one of his things where, oh my God, it's a 10 petabyte mechanical supercomputer. And no, your brain is not a supercomputer. It's not a computer at all.
I mean, you could make it. Okay, that's.
You can make a case for it.
Well, hold on. What do you mean it computes nothing? Because because you take an input stimuli, and you come out with output.
Yes, like I say, you can make a case for it, but it works nothing like a computer. It doesn't work like a computer. Let's put it that way. Our computers are very, very different from how your brain works.
I think you're taking the analogy too literally.
The analogy too literally. Yeah. It's, you could say it does computation. It sees a pattern and it goes ahead and, computes that pattern, comes up with, midnight's in the fucking room again, bothering me. So I have this midnight neuron that lights up. So anyway.
You just have to take a shot at me, don't you?
I do. You know what? It's for your own good. It's because I love you. I'm trying to strengthen you.
I have something curious to contribute to the conversation.
Yeah, go ahead, Scott.
No, computers, for everything they do, literally do calculations. They do bit shifting. Which is a form of calculating,
right, which your brain doesn't do.
The brain doesn't do that. It's not a literal table of calculations or truth tables or whatever
right.
For every process, for every action.
I mean,
there's no code. There's no higher level language that your brain is running.
It's more like it's like a huge string of spaghetti with little notches on it at the best, just kind of running an analog kind of system.
I could make a case that a river. Oh shit, cat! Come on, that's my fucking microphone. You can't rub on my microphone.
Thor just has no respect.
No, he has no respect for the microphone. He tried to pet himself on my microphone, and it flips over. I could make a case for a river running in a particular pattern as being a computer, right? It's computing its path as it goes along. I mean, we can make all kinds of analogies. That's what the brain does. Brain makes patterns. Brain finds, condenses things and makes patterns. So anyway, how many papers have you read today, Dessel?
300. Oh, wait, you're talking to Dessel, not me.
Yeah.
None so far today.
Really?
Yeah.
Wow.
No, see, Dressel, you should have agreed with my number.
300, I'm working on it. The only thing I really had for you guys was for yesterday's stream with on Ger's stream that went for like 10 hours. I think Otangelo misspoke. That's what I'm going to go with. He was in a conversation with Immutable about metabolism first approaches to abiogenesis, and he said that Robert Shapiro refuted metabolism first approaches over 10 years ago. But that was in the context of them already discussing abiogenesis and coming from it. I think it was Maddox who mentioned RNA world, so they were kind of going in that context. So I think Otangelo meant to say that Shapiro refuted, quote unquote, the RNA approach, not metabolism first, because Robert Shapiro is a metabolism first researcher, or was when he was alive. He supported metabolism first. So I just thought that was weird that he was saying Shapiro refutes metabolism first. But
Yeah.
Why couldn't RNA and metabolism appear at the same time?
Okay,
Well,
one more thing. One more thing about that, Dessel, before we address midnight. He didn't refute it. That isn't what scientists do. They don't fucking refute each other. They come back, criticize each other, basically come up with a new hypothesis, kick things around. I mean, when did they ever use the word, I refuted you in this paper? Is that fair, Dessel?
So, I mean, that's something that if you were to talk to maybe Danny or Leo or maybe James Fodor over on the Digital Gnosis channel, you'll get a more warm reception to that kind of idea. But the way that I understand science is that it's more substantive than a lot of people want to say. Like, I don't even object when people say that science proves things, which is a very unpopular thing to say right now. But as far as I'm concerned, it does. It always has been in the business of proving things, and it always will be. But I mean that in a particular context, in the context that we would refer to in the court of law, when we have this standard of proving something beyond a reasonable doubt. That's what I mean by prove, and that's what I think the average person means by prove. And so, can a scientist refute another scientist? I would say yes, if they bring conclusive evidence to the front that shows that their idea can't possibly be right by any reasonable standard, then I would consider that a refutation, a substantive refutation.
So, do you think that Robert Shapiro single-handedly shut down the entire RNA world hypothesis?
So, I mean, I just got my hands on Shapiro's book Origins that goes all the way back to 1987, The Skeptic's Guide to the Origin of Life or whatever. I don't know the details of his particular case, other than what it says here in this article is basically him focusing on the complexity of RNA. That it's not so easy to form, which is something that origin of life researchers spent a long time on, is nucleotide synthesis. It can get tricky. But I would actually agree with Shapiro in terms of of it being RNA only, because I like the metabolism approach where you do end up having them both together at the same time. And what kickstarts that process is a rudimentary metabolic pathway, like the acetyl coenzyme A pathway, that you have going on at hydrothermal vents. So like the metabolism would kickstart it. You end up in a situation where in the same environment, you have amino acids and nucleic acids. They concentrate and polymerize and stabilize each other and start interacting in that particular environment. And eventually you get an overall coherent unified structure out of that. That's how I would understand it. I think that Shapiro makes good points in that regard. But the way that Otangelo, though, he gave the chicken and egg problem, which the RNA approach was originally designed to overcome because of the capability to act both for catalysis and for replication.
Ribozymes, yeah.
Well, for like RNA's ability to both operate as something that can catalyze reactions and something that can replicate. And so that's how they were trying to solve chicken and egg via the RNA world. But from my perspective, you don't have a chicken and egg problem because you have them both there at the same time. So there was never a time when a chicken was first or an egg was first. They came hand in hand together through time.
Right.
Sorry, I'm not laughing at you. I'm laughing at something else.
What came first, bacon or eggs?
Eggs.
I don't know. Anyway, I think it was bacon.
I need to go.
Okay.
I'll be back. Hopefully.
You can't stand it, can you?
No, it's not. Okay, look, but Dressel, did you address what I said or asked?
Yeah, I think I just covered it in the last thing that I said there, that they are there together at the same time. That you only really get a metabolism first in the sense that a metabolic process or cycle kickstarts it. in terms of what's going on at the vents, say, if you're going for alkaline hydrothermal vents, that would kickstart the process, but you end up with both peptides and RNA in the same place. And so there was never a time, it was never the case that, okay, go ahead.
I know. I do have a question. Considering there are so many possible ways life could have started on this planet, are we ever going to determine the one way it started?
In my personal opinion, I think that we can get there over time. There are ways to eliminate different competing hypotheses based on the data that we have or will have. And I think that we can get pretty close already. Like I said, I'm really optimistic about hydrothermal vents overall. So that's what I want to say is the way that life originated. And I think that there's good evidence for it. But, it is going to take a little bit more time on my part to fully make that determination with full conviction. I still want to continue to research, but that's my research program because I'm optimistic about it.
Yep.
All right. Anyway, so I don't know. Speed. If you want people to come in, you should really pin. You should really pin the link.
I don't know if you can pin. If I can pin anything, I don't know how to pin things.
I'll say one last thing, and then I'm going to shut up because I want to address what Professor Dan's saying in the chat about as long as it's not a understanding of proving something that is according to a strict standard of mathematical proof. And yes, that's the difference. If you're just going to have that conception of prove where you have to be able to put QED at the end of your statement, then yeah, there would be a lot of science that doesn't prove anything because your understanding of proof is just limited to the realm of mathematics. But my argument here is that there's another equally substantive notion of proof that we already have well established historically in our court system, which is the only real system, like the only real standard that really matters to us on an important practical level, on a day-to-day level, is proving something beyond a reasonable doubt, proving that, in your day-to-day, when you ask questions or trying to find out the truth about something, that's the standard that you're operating on. You're not putting QED at the end of your every day-to-day statements.
Fair enough. Apparently, nobody gives a rat's ass. Anyway, so come on in, Andrew, Dr. Dan.
I care about you, Dessel. I care about you.
You care about Dessel, yeah. We do care about Dessel.
Dessel's just so awesome. I was just stunned. I couldn't move my mouse.
Yes, it was stunning. Yep. Oh, so I'm catching bugs tonight.
Yeah.
Yeah, because it's hotter than hell out there, so it should be a good bug night. But I wonder if I shut my east side lights off, if I would attract more bugs to my light. I bet I would.
What's your temperature there now? How warm?
I don't know. It's gotta be
60. I'm 60 here.
Oh, we were in the 80s just a little while ago.
Yeah, we're still 88.
Yeah, I think I was only in the 70s today. I think even low 70s, I believe. Actually, I'm not sure.
Updates if I click on it.
Let me see what it actually. Yeah, I think today. Yeah, it was low. I was low 70s today, so not really that warm.
It's 82 degrees here right now.
Really, that hot, huh? Wow.
Yeah, and it's windy. It's weird out there.
20, both be
22 degrees cooler.
So when are you going to be in New Mexico, Dessel?
My bus is on Tuesday.
Wow.
You're taking a bus like a Greyhound?
Yep, that's how I always travel.
Tell me you'll have a car strapped to your back.
Oh my God! Is that like a covered wagon sort of situation? Or they still have those, eh?
Yeah, I think some of them are still led by donkeys and stuff.
Are they? Well, be careful. I've gone on bus trips and I've ended up with some really bizarre fucking experiences. Like really bizarre experiences. Just be careful out there.
Yeah. That's how I've always traveled. I've jumped all over this country on buses.
God damn it. Scotty.
Finally, Orge shows up.
Hola.
Okay.
Hey, Orge.
Hey, I don't know how long I can spend. My kid has been restless recently, but I'll do what I can.
You know,
Did you go ahead.
you know, we've had a week where we discovered that duct tape might help.
Yeah.
Yeah.
You can thank David for that.
Yeah, David, by the way, duct tape works both ways.
I know. Well,
Yeah.
but I figured I'd probably go to jail if I did it the other way. So,
Well, shouldn't she go to jail? I mean,
yeah,
toddler prison?
yeah, exactly.
She kidnapped you.
I'm telling you, she almost I almost was trapped.
Yeah.
Like, seriously. If I let her go, I just barely stopped her. I was completely, one arm was on there. If I let her do the other one, I would have been screwed.
That's like back when my daughter was a little. I had my friend Mac. He was like a real peaceful, just almost unresponsive person. He really brilliant, but he just had a real easy style. So we wouldn't have been surprised if he would have let her. He would have let her duct tape him to a chair. But we came home one time, and there were all these axe marks on the windowsill, and it's like, what the hell, McCormick? And it's like, oh yeah, Faye was chopping the windowsill with an axe, but I was making sure she didn't get hurt. And it's like, okay, your job as a babysitter goes a little bit further.
Yeah.
It's like, no, he just sat there and watched her chop the fucking windowsill. It's like,
Yeah,
okay.
yeah.
As long as the kid doesn't get hurt, as long as she's having fun, it's like, it's okay.
That's kind of what I do.
Yeah,
I mean, not to
to
that extreme, but yeah. We do some fun things.
Yeah.
Got to keep it entertaining.
Yes. So, Orge,
Yes.
do you know anything about dating methods? Potassium.
People? I know how to pick a lady up at a bar, if that's what you're asking.
No, that wasn't where I was going.
Sorry.
Yeah, I was talking about argon ions, and basically, Standing for Truth has this chart he puts up from contested bones.
Contested bones.
Yep, it's
Again, that sounds like the original topic here.
Yeah. So anyway, he puts up this chart and he says, why should we trust radiometric dating if they get all these things wrong? And if you go to contested bones, they have a chart, and they apparently use the wrong column. They use the ratio of the isotopes as millions of years instead of using the column on the right. So they didn't even use the right column. And the paper was about these recent, historic volcanic eruptions that we know about. And the paper was about how we have to be careful with argon dating because of X, Y, and Z, right? And the paper was all about what went wrong, right? So that is their example of why we shouldn't trust any radiometric dating. But the weird irony here is that the paper was primarily scientists that do this radiometric dating telling you how to do it right.
That is such a common failure of communication about this topic between Yex and the rest of us.
Yeah.
There are things that radiometric dating does not do well, so you don't use it. Radiometric dating becomes flawed if the thing's 5 minutes old because you got too much shit going on. Radiometric dating doesn't work if you use the wrong dating method. If you try to use the wrong elements, it doesn't work too well. Like, there's lots of things that you don't use radiometric dating for, but Yex just find known issues and say that's somehow true for the entire thing. It'd be like trying to cut a steak with a fillet knife. Like, you're going to kind of fail at it. That doesn't mean you can't use knives to cut steak.
Yeah, and they say that experts found, the experts in the field found flaws in the field. And as it turns out, that's what the experts are pointing out so that you don't make flawed measurements.
Yeah, I've had this conversation. I think 2 or 3 of my videos on this subject have been exactly that. What you're pointing out as a flaw with the system is exactly that. It's a flaw that you work around. That's the whole point.
Yeah. A flaw noted to make the system better.
Yeah.
It's just like, yeah, but SFT puts up this table and he's put it up like at least twice in the last 2 weeks. And he's like, well, why should we trust anything you say? Because this. and he shows this table.
I don't know what this table is. I'm looking for it, but I'm not finding it.
I've covered it in Friday's weather report, I
I
believe.
can scan through that real quick.
Let me check and see.
Okay.
And I also listed the sources. Let me. Okay. Let's see. I think it was on the 17th. 10 more answers. Babylonian exile. 7 life lessons. Eric Lerner. The biggest nope. I guess it might have been Thursday. Okay, let me go back to Thursday and see if I can find it for you. The 17th, that would be the 16th. Let's see, talking with William Radcliffe, dark matter. I have no idea about time anymore, do I? When did I cover that?
Time?
No, when did I cover that geological thing?
I don't know. You talking about the phosphate beds?
No, well, we were talking about those two. No, that was last night, right? No, I put up the papers, Common Ancestry of Life, Simon's Funk. Oh God, did I not put it? I'm sure I put it in the description. Okay, Darth Ron's wavelengths. I'll track it down for you. You guys talk.
Quick question for Dr. Dan, and then you guys go ahead. But I was just wondering whether or not Dr. Dan has engaged in much Bayesian phylogenetics, and that's it.
I wonder if that's something that they get computers to deal with and just crank out a number.
Oh, I lied. There was another thing. You just brought up contested bones. Did you watch like the 8 hours worth of content over at Gutsick Gibbon covering a lot of the material on contested?
Oh yeah, I did. I was just trying to. For some reason, I thought Creation Myths had a video on it, and so I was trying to get him to plug it, but he may not have. I may be misremembering.
Heard.
But yeah, I mean, a lot of that shit from those books has been thoroughly debunked by either Gutsick Gibbon or Creation Myths, or you have R.J. Downard.
Well, that's the weird part about this whole conversation is you don't debunk it. It's just part of the conversation. Like, you can't debunk that these flaws exist. You just need to explain that the flaws are part of the process.
Yeah, that
and that conversation is like never actually happens.
It's just one of those convenient, or should I say for them, inconvenient truths that never get discussed.
Yeah. So the idea that their God is somehow flawless, so therefore all science needs to be flawless too. It's ludicrous.
What's up? How are you running?
I can't find. I went through it to the 7th. I can't find it either.
You're looking at the 17th?
I went all the way to the 7th.
Yeah, shit.
What's wrong?
What did I do? Did I talk about it and not put the links up? God damn it. Okay.
So as I've said before, on my master's thesis, I did utilize radiometric dating to confirm my thoughts on it via my geomagnetic dating, and they turned out to be accurate. So I've used it. I sent it to the lab and it came back, and I read the reports and things were accurate. So I know this tool works. And had I used a different radiometric measurement, I probably got different answers because I was using a different tool. So I mean, it's
well, in those cases though, aren't there like error bars, and even if you use like different methods, they should pretty much converge within their own respective error bars. Am I making sense?
But I mean, error bars are exactly that. I mean, once you get down to the far ends of, say, carbon-14 dating, the error bar gets so huge, you're kind of pointless to use it.
Yeah, exactly.
I mean, I think I used potassium argon dating for my thing because it was 1.5 billion or something like that. But had I used like helium dating or something like that, that's in the same age group. But then again, you have the mineralogy of my samples was different, so I couldn't use helium dating, and all this other like things that people that use this stuff accept as par for the course when you're doing radiometric dating, but somehow a creationist thinks is a red flag or flashing red light. It's impossible.
Well, yeah, but I'm sure they think if they can frame it that way, they've got a propaganda tool. So
yeah. When a scientist doesn't think of it as a propaganda tool, we think of it as a tool.
Yeah.
Because we're trying to answer questions, and they're trying to not answer questions.
Okay, so Jorge, I put the link. Oh, okay, Contested Bones is the book, and I'll get a link for that. But I just put a link for the paper. What happened to my link? For the paper, okay, the Contested Bones refers to a paper by Dalrymple or something.
Yep, the argon analysis of historic lava flows. Right,
that's the one they refer to, and they copy the table, but they use the wrong goddamn column for the dates. Instead of using the column on the right, they use the one in the middle, which is the actual ratio. The dates are on the right. And it's kind of funny that they do that because it shows you how shoddy their work is, right?
Yeah, they're bad at quote mining.
You know, unless I got something wrong, I'd love you to look at it and make sure I don't have something wrong on that. But I looked at the damn thing, and it's like, okay, where is it here? Hang on a second. Let me show you. Dalrymple? Where's Dalrymple? Oh, God, for God's sake. Okay. Let me share it.
Of course, Dalrymple, whatever the heck his last name is, was not the person they were talking to about this. It was third and fourth parties.
Yeah.
So here's
the paper. Okay, here's the table that they use, right? So okay, the let's see, the Hulalai 1800-1801. If you go over here, okay, they say that what the hell was it? Sample weight, okay. This isn't the table. It's further down.
Yeah, that's measurement.
Okay, this one, this is the table right here. So Hulalai is 1.6. They say it's 1.6 million years. And over here on the right, they have apparent age as 1.19. So they use these numbers, which I think is the ratio, how much argon there is per mole or whatever. Something like that. Okay. So if you look at their table.
How many moles per gram? Yeah.
Right. So they say it's 1.6 million years. For Mount Etna, I think they had 0.25. And they use the wrong column. But take a look at it and see if they really did it. I'll find Contested Bones for you. Oh, let's see.
Yeah, what I'm looking at, the second and third columns are not ages, and the fourth column is
apparent age.
Apparent age, right? In 1.19 million years. But as I said, without I'd have to read. The chart doesn't tell you. You need to read the paper with the chart in order to understand the chart better. And yeah, I mean, if you could link me this paper, I could scan it real quick. Although your scan of it probably would pull the same information mine would.
I did link it in the public chat.
Okay, I'll try again.
Okay.
I wanted to just real quick. I'm sorry, it's just an irrelevant topic to what you guys are talking about, but quickly to thank Dr. Dan for answering me in chat, and to say that I originally got excited about Bayes' theorem in philosophy, in epistemology, and the philosophy of science as a way to answer the problem of induction. And the pioneers of doing that work, people like Richard Jeffrey and then later Colin Howson and Peter Urbach, they provided a formal foundation for inductive inference. So doing really well in terms of answering that problem of induction. But it was also really awesome to see it get into phylogenetics, so ending up with practical applications as well. And I wanted to say that because I know that Dr. Dan has had to deal with a lot of philosophers that kind of ruffle his feathers because he doesn't think that they make any good contributions to the sciences. He might see a lot of philosophers as basically sitting there in their armchair dictating how he, as a scientist, is able to do things without having any actual feet on the ground in terms of science. But just to reassure him that there are some areas of philosophy that give him useful things, such as Bayesian phylogenetics. And I've been reading some papers on it recently. And the relaxed clock models seem to be performing in a much more superior way to some of the more older approaches I will find the paper that I'm talking about on this. But yeah, so I mean, I was wondering. I'm interested in Bayesianism in general, and I really like to see how it's being applied practically speaking today, and how successful it's being with respect to that.
I think I just found the paper to download to go back.
I put it in the description too, Jorge. So it's in the description of this video. But I have their chart here. I've got the Kindle up of contested bones. Now you'll see what the hell is it? The column at the end here says apparent age is 1.19 million, and the ratio is 1.6 plus or minus 0.16, right?
Yeah.
So now if you go to the book, let me share the book. Mike's Kindle.
Book. Show us the book.
There's the book. Okay, the book says, can you read that?
Let me go full screen. There we go.
Shit. Oh, shit. What the hell did I do with the...
What did you do?
I lost it. Oh, okay. There it is. How the hell did you...
Mount St. Helen readings.
Oh, my God.
What is that what you're doing? I just saw a mention of Mount St. Helens in the text.
Okay, you see they say 1.6 million years here?
Yeah.
Yeah, and from the chart, it was 1.19 million years. So they used the first column, the ratio, and they did it over here too, Mount Etna, 0.25 million years ago. That's the ratio. The apparent age was different in the paper. So, I mean, like, they didn't even copy the fucking table right. And this is from 2017. The paper is from 1969, I think, or something.
It looks that way, yeah.
Kelly Rimple, yeah. And it's like, oh my God, guys, get better, do better. It's like they used the wrong column, I think.
I am being yelled at by my kid, so I'm going to bounce. I can come back later if she happens to go down. But yeah, from what I'm reading, it looks like you're correct. I'll try to do a bit more digging, see if I can do any better. But yeah, I gotta leave.
Okay, 2 words: duct tape, sir. Duct tape.
No, terrible people.
Tape.
All of you, terrible.
Also, oxycodone works on toddlers like a charm. Just fucking excellent. But do not use it for more than 2 weeks because they may get addicted.
Okay, what's this codon bias that they're talking about in the site?
Oxycodone bias?
Maybe oxycodone bias. Is that what it is?
Yeah.
You're on drugs, Andrew?
Creationist, does codon bias lend any credence to the shared feature, shared function nonsense? Okay, codon bias. What are you talking about? Are you talking about...
I asked Andrew to come in and talk about it, but...
Are you talking about the wobble at the end? What the fuck?
Wobble, wobble, wobble. Sorry, that was a dumb...
Okay, are you talking about the effects basically of ribosomal pausing and particular codon sequences? Codon bias, because I will always talk about codon bias.
But Andrew or Creationist, we need y'all to come in and talk about
codon
wobble
bias
and codon bias.
Yeah, because the thing is that codon bias frequently works as a regulation mechanism because the ribosomes pile up and then they kick out the mRNA and they quit producing. But Maddox wants to make it look like a 3D printer. He believes that it's like this amazing piece of coding, and it's actually more regulatory. Occasionally, they found instances where ribosomal pausing will allow a protein to fold more easily. But most of the time, I think it downregulates the protein synthesis. But I don't know. Dr. Dan probably knows more about that than I do because that is a prokaryote topic particular. And that is, I think, his specialty. No, viruses. Anyway, give me 30 seconds. Dr. Dan's coming in.
Hey, guys. What's up?
Hi, David.
Hey.
You just had a stream, right?
No, I'm not. I rescheduled the stream for 10 o'clock, so in about 20 minutes or so.
Oh, shit. So we're going to overlap you?
Immutable, how are you doing?
I was running late, so I guess it all worked out in the end because I didn't want to overlap you guys. So if need to, I can always bump it up a little bit. I'm going to be talking with Jordan about some of the recent bullshit from Matt Slick, and it is a doozy.
Okay, good.
Oh, bullshit from Matt Slick, huh?
Yeah.
Good. So you give me an
Forward to that.
excuse. So you were planning on starting in 15 minutes or an hour and
20
50?
minutes, 25 minutes or whatever.
Okay, you give me an excuse to shoot this in ahead early. That's great. Thank you.
But not until Dr. Dan comes in and talks about codon bias.
I want
Right.
to hear Dr. Dan. Feel free. I don't want to cut everything short until Dr. Dan comes in. 'Cause I love-
Really, Andrew needs to get on because he's going to want to ask questions about something.
Yeah.
So,
Yep.
Notable, what are you doing that's new?
Not too much. School is just school, and I haven't been able to read too much, so all the fun shit.
It's wearing you out, huh?
Hey, Dr.
Oh,
Dan.
yeah.
Good to see you, my
It's killing
friend.
me.
Hey, y'all.
Hey, Dr. Dan. Yes, we do want to talk. Yeah, to answer your question, yes, we do want you to talk about codon bias.
So before you do, Dr. Dan, note that Daniel Bennett is cautioning you to remove your bias against God, and then the codons will come into line.
I'm sure.
Okay. I'll keep that in mind.
Yep. Okay.
Your mic is like near your ear instead of near your
No,
Yeah, yeah, but-
I got a different mic. This is
Oh,
okay.
I'm not using this. Yeah, I'm not using it. Yeah, it's weird. Everyone gets confused because I have this thing, but I'm not using this mic. I'm using a different. Yeah, it's fine.
Hello, Andrew.
Now Andrew comes in. We should throw him out.
Yeah, I told him to come in because he's going to want to ask questions.
Okay,
Oh.
codon bias.
You're talking about codon bias. That's amazing that there are, like, raise your hand if you care about codon bias, because there's approximately 15 people in North America that care about codon bias. And I'm one of them, and my graduate advisor was another one. And, like, that's a large percentage of those people right off the bat.
Good.
So, okay. So here's the thing with codon bias is that so I'm all about viruses. That was how I looked into it. And a very, very specific group of viruses is what I worked on with codon bias. But the big idea with codon bias is that except for some very specific circumstances, it doesn't matter that much. Like, selection for different codon usage profiles is really, really weak, to the point where if you have like a biased mutation rate, then that's going to dictate what codons you use rather than like what the tRNA pool prefers in a given organism. It's
Okay.
yeah. There are some exceptions. Like at the front end of the mRNA, you do have some preferences because the first like half dozen to dozen codons are going to affect the initiation rate. Like
Right.
when you're recruiting all the stuff to start translation. So the first handful are going to matter a lot, right? You got to throw those out. If you want to know like what like the baseline codon profile is for an organism, you got to throw out like the first like 10 or 12, because those 10 or 12 are going to drive the rate of initiation. So they don't matter as much. But then for the rest of the mRNA, I shouldn't say they don't matter. They matter, but they matter in a different way. They're not trying to like match the tRNA pool specifically. And then for the rest of the mRNA, you're striking a balance between translation rate and accuracy. So the faster you are, the more likely you are to have mistakes. But if you're super slow, then everything's inefficient and you don't work as well. So you have to find that Goldilocks zone where you're going as fast as you can while minimizing mistakes. And that like kind of selecting for that exact right, like which codons you use is pretty weak, as at least in viruses. I can't speak to other organisms, but I'm really familiar with it with viruses, and it's not a strong selective pressure at all. And to provide data for that, if anyone wants the references, I get to cite myself for once, which is cool because my papers that I've published are on viral codon bias.
Very nice.
Now,
we have an expert, guys.
In eukaryotes, I believe I found some papers that indicated that certain codon sequences will slow up the translation process, and it will downregulate the protein, basically.
Yes,
So-
that's
so-
absolutely the case. Yep. Mhm.
Okay.
Now, generally, you're going to use, because eukaryotes are different from prokaryotes, and that for prokaryotes, because think back to your basic cell bio, everybody. Remember your high school prokaryotic versus eukaryotic cells? Your prokaryotes are all happening in one place, right? So you do transcription and you do translation, and it all happens simultaneously. So, like, once you start the process, it's just going to go at whatever rate it goes. For eukaryotes, everything's happening in different compartments, so you can regulate the rate of each step. So you can do transcription at one rate, and like you can go like all out with transcription, but you can still heavily downregulate that same protein by hitting the brakes on translation for those mRNAs. And codon bias, it plays a role in that, but it's not. It's a lot less important than things like translation initiation is a much bigger factor. The length of the poly-A tail is a much bigger factor.
Yep. Yes.
Because that regulates the lifespan, the half-life of the mRNA, right? So I don't want to say codon bias doesn't matter, but in eukaryotes, it plays a much smaller role compared to the other things. Does that make sense?
Yep. Maddox would be very disappointed to hear that.
I've talked about this with him, unfortunately, and he did not care that I spent 6 years in graduate school studying this exact thing. He did not care. Surprise.
He's so brilliant that
like,
nothing matters to him.
Like I literally, I'm not exaggerating when I say I literally wrote a book, and half the book is on this topic, and like just doesn't care.
Question is,
so like I don't know.
So back to the other question: do you think FedEx is just dishonest or incompetent or both?
Why not both? Why not both?
What was that?
I said, why not both?
Yep.
I think he believes in himself, perhaps too much.
If he believed in himself so much, I mean, I've invited anybody who has specifically criticized me. My standard response is, thank you for your input. Please come on and we can have a conversation about this. And so far, nobody has taken me up on that, with the exception of, to his credit, Otangelo came on for a conversation about irreducible complexity. Nobody else has done that. Nobody else.
But Otangelo is like an impenetrable wall. Yeah. It's just like a barrier.
But to his credit, Otangelo doesn't like call people like rude names in conversations.
He's been getting pretty close lately.
Oh, is he getting closer? Because I mean, this was a while back we talked, but and then once classes and stuff, I get busy.
His transformation to the Darth side is complete.
Yeah, he's changed a little bit in the last few months.
Oh,
unfortunately,
that's when I've been kind of not as paying as close attention. That's sad to hear.
Yeah, it's amazing. After a while, the ravages of YouTube get to some people and they become nastier and nastier. Hopefully it doesn't happen to all of us. Well, it happened to me. I mean, I was a softer, kinder, gentler speed of sound when I started this shit, and I just got meaner and meaner as I went along.
I can't imagine why you would get more cynical over time interacting with younger creationists on YouTube.
Oh, yeah, right. I can't imagine.
I don't know why you would...
I think the word is frustration. It's just, you keep beating your head against the same wall long enough and you're going to you're going to change your personality.
Yeah, your pupils are going to dilate unevenly.
Yep. Yeah.
So what's like the whole argument with codon bias? Like, why is that a whole thing that came up?
Well, he's trying to make the case that there's this strong programming element in mRNA. That it's so highly programmed that it creates this 3D printer, that sort of thing. That it's just that it's really complex and it's multiplexed and it has many layers, and that's just one of the many things that he uses to prove that there had to be a coder because this code is so perfect.
Can I give an example of something going back to what Dessel was talking about earlier with the less stringent definition of prove? One of the things I'm proudest of is that, like, my graduate work, I actually explained, I think I proved what was going on with codon bias in a specific group of viruses. And the way I did this was looking at genes that were encoded in opposite directions on the genome. So it's a single-stranded genome, which means instead of being a double helix, it's a single helix of DNA, right? It's single-stranded DNA.
Yep.
And they have two big genes, like one on each side of the genome, but they're encoded in opposite directions. So one is the positive sense, meaning you need to make a DNA copy of it and then read mRNA off of that. And the other gene is encoded in the negative sense, meaning you could directly read mRNA off of the gene, right?
Okay.
And so what we know about these viruses is that their codon bias is weird. It's biased towards specific sets of codons. And what I did is I looked at those two genes separately and said, okay, if it's selection for like a specific set of codons, like the host cell is imposing a selective pressure on codons, then in the genome, they should look different, right? Because they're encoded in opposite directions. But then in the mRNA, they should look the same. But if it's not selection that's driving it, if it's just mutations that are driving it, then in the genome, they should look the same, but the mRNA should be opposite. And I looked at hundreds of different viruses, hundreds of different genomes on GenBank, and put all this stuff together. And wouldn't you know it, in the genome, they were opposite, but in the genome, they were the same. No, I had it back. In the genome, they were the same, but in the mRNAs, they were opposite, which meant the virus didn't care, relatively speaking, about the tRNA pools, because one mRNA for this for one of the important proteins used one set of codons. The other mRNA for the other important these viruses have two major genes and then a couple smaller ones, but it's really just the two. So one of them used one set of codons. The other mRNA used the opposite set of codons that completely reversed codon preferences. But in the genome, in the single-stranded DNA genome, the wobble sites were all the same, which I'm going to use Dessel's definition of proof, which proved that it was the mutations that were in the single-stranded DNA. You have specific types of mutations that were more likely than others. So you had this biased mutation rate.
Right.
It proved that the biased mutation rate was driving the codon bias, not selection for efficient translation in the host cell, which just goes to show you how weak that selection is. So anything Maddox or anyone else wants to say about, oh, it's this, it's that, it's fine-tuned, like, no, it's weak selection. It's not strong selection.
Yep.
Thanks for that, Dr. Dan.
You're welcome.
Okay, real quick, I want to plug something since we're talking biology and stuff tonight. Cover your ears, Speed.
Oh, no.
On Friday afternoon at 4 p.m. Central, on the Phil Talk YouTube channel, Danny's going to be talking to Dr. Daniel J. Nicholson, a philosopher of science, a philosopher of biology. And Nicholson, I think, is very underexposed on the internet. He gives some good papers on, how we should look at life, how we should look at the cell. He also did a great talk, and I'm going to put another thing in the private chat. We've done the link to it before. I'm talking midnight. It's called "What Is Life?" A retrospective evaluation of that. The name of the "What Is Life" is a book by Schrodinger about the cell, about how it changed the field of biology. It's an excellent lecture. It starts out Spanish language as for like introductory stuff, but then it goes into English and everything. But if you haven't seen that video, I just encourage everybody to go out there and watch it. I found it to, of course, people who are versed on the history and philosophy of biology, maybe that, that's just old news to them, but I found it to be very fascinating and enlightening. And so you get a chance to hear Daniel Nicholson talk on Friday about, how he feels about things like that. And I'm sure he's going to be asked about Jerry Fodor.
It's not only
where's the link?
In private chat.
Okay, the link is in the description. So Nicholson, unlike a lot of philosophers of science, he actually knows a bit of science, and he pushes back on the whole cell as a machine. he pushes back on the coding ideas, the analogies, all of that stuff, and he pushes back on it heavily in all of his papers and lectures, and he pushes back on what Schrodinger did to biology, basically setting us on this course of looking at it as a machine. And so that's kind of Nicholson's big thing, I believe, is to push back on it. Yeah, I think you'd like Nicholson, Doctor, and he seems to know quite a bit of the science as well. He doesn't just spout bullshit like Fodor. So.
Okay, good. Yeah, because I read that book and I'm not impressed by that.
What is life you read, or the Fodor?
No. Fodor, the problem with whatever it was about Darwin.
Have you read What Is Life?
I have not, but now I'm adding it to my list because that list isn't long enough.
Anyway, that video on the What Is Life book of Nicholson is in the description along with something else. Yeah, so anyway, check it out.
It's open. Yeah. It's right there, so I'll get to that.
Do any actual biologists take Fodor's argument seriously?
I don't think so.
Can I rant briefly on the extended evolutionary synthesis before I go?
Please.
Okay, here's the thing with the EES. So there's kind of two sides of it. One is a bunch of mostly not evolutionary biologists and frequently philosophers and philosophers of science telling evolutionary biologists how they need to do their jobs. But here's the thing that they don't understand because they haven't looked into evolutionary biology. The stuff they're saying biologists need to include has been part of evolutionary biology for decades. So it's neutral theory, evo-devo, niche construction, genomics, multi-level selection,
multi-hierarchies.
Right. All this stuff that's been part of evolutionary biology since the 1960s. I mean, if you read Fodor's book, it's all about the modern synthesis the modern synthesis that. Dude, the modern synthesis is from the 1940s. We didn't stop in 1948. We didn't just stop doing evolutionary biology. We've been doing stuff since then. And so they act as though the field stopped at this point 75 years ago. And there's no recognition that the field has advanced beyond the point where Dobzhansky was a big name in the field. It's we've been doing science since then. So that's the first part. And then the second part is if you've ever checked out the third way of evolution people, there's a pretty significant overlap between the extended synthesis people and the third way people. But the third way people also get into some really wooey stuff like cellular intelligence. And there's just nothing to that. It's complete hogwash, cellular genetic editing or something that cells can manipulate their DNA to get the phenotypes they want. And no, none of that is valid. That's not how any of this works.
On the other hand,
everything about it is wrong. It just makes me so mad because.
On the other hand, there have been some books written that have talked about extended synthesis that do have a whole lot of valid information in them and hypothesis, valid analysis of what the state of the art in biology is. I think. Yeah,
one of the papers that I really like on that topic is Dennis Noble 2015. I use a figure of his in class. He calls it the integrated evolutionary synthesis.
Yeah,
I modified a figure of his that I use in class that I've now come to learn my version of that figure is floating around the internet. Jackson Week used it in a video before I met Jackson. So then I met Jackson and I was oh, this is,
serendipity or something. But by the way,
and then I was looking through his videos and one of his thumbnails, I'm I made that figure. This figure is floated around the internet and I think that paper is a fairly fair overview of the field, but what a lot of the EES proponents don't get or don't acknowledge is that the stuff that they're promoting, we're not ignoring it. You could go through stuff that Fodor says in his book that evolutionary biologists don't take into account. And it's stuff that I teach in a 200 level evolutionary biology class. And if I'm teaching it in a 200 level class, it's not cutting edge stuff. It's well established stuff in the field.
Yeah, he
So,
focuses.
what are you talking about?
Do you
focuses
remember the
a
name of that paper, Dr. Dan?
I'll look it up and I'll put it in the I'll put it in the I'll put it in the public chat. Just give me a second.
Okay.
I'll pull the Noble 2015 one.
Yeah.
He
Yeah, I'll find
focuses
it
a
real
lot.
fast.
He focuses a lot on the earlier parts of his book, all on Evo Devo. So yeah, exactly what you're saying. And I just want to put in there too that they do the same thing with abiogenesis. I
It's
hear
the same
constantly.
thing. Yep.
Yeah, like
Mhm.
they just act like nothing's happened since the Yuri Miller experiment.
It's right, exactly. And it's like, dude, we've been doing so much research. And like I think you said earlier about how hydrothermal vents are super promising. And like, yeah, and that's cutting edge stuff. That's happening like year to year right now. But like, if you're going to pretend the science stopped in 1953, then like, I can't convince you that that's wrong.
Wait, yeah, like Otangelo brought up yesterday, Robert Shapiro. And I mean, like Shapiro, his main book was all the way back in 1987. He did have a 2008 thing, but still, even on some of the streams that I've been watching recently, you can hear explicit quotes from people saying that it hasn't progressed at all in 70 years.
Like, what? Are you kidding?
That paper's
I
also in the
think-
description.
I think, Dressel, I think you just broke Dr. Dan. He's finished. He's done.
No, it's like, what do you say to that though? Like, for someone who says the science hasn't advanced since an experiment that was done, 50, 60, 70 years ago, like,
Yeah.
how do you convince someone that that's not right? Like, I could cite all the papers in the world, but if they're not going to believe that we haven't done anything since the 1950s, like,
Well,
I'm not
Dr.
gonna convince
Dan,
them
Dr. Dan,
otherwise.
Dr. Dan. That's when you walk away from, you walk backwards. You're like, okay.
Really, it's true. No, it's true.
So I've got three papers by Noble, 2010, 2017, and 2021.
I just put
Is it-
put the 2015 in there.
Oh, you did? Okay.
Yeah, and it's I'll actually link the figure that I made. It's figure 1 in his, but I adapted that. So I'll throw the link to my version of that figure in there because I made it smaller.
I've got this. I've got that paper out in the Horseman directory, but it's mislabeled as 2010. So
Yeah,
let me
no,
fix that.
that is.
I really
worth
gotta get
reading.
back. I really gotta get back reading reading the epigenetics books in the book in the Horseman Drive.
Yeah, you do, instead of torturing yourself on Discord.
Fair enough.
I'm just saying. I consider myself your sponsor, so.
Well, you know what, Speed? I appreciate it.
The paper I was talking about before on Bayesian phylogenetics and the greater accuracy of some of the relaxed molecular clock models, that paper is in the private chat. It is a little old. It's from 2012, but nevertheless, I think it's just explaining what has just become like a increasing trajectory for Bayesianism in phylogenetics. So
I think that's fair because as we look at like so what I did in grad school was looking at like base specific mutation rates, which is why I love codon bias, because cytosine mutation rates drive codon bias in single stranded DNA viruses. There's a fun fact for you. That nobody cares. No one cares. But
I do. I care.
What I was doing was looking at like base specific mutation rates. And so if you're doing your phylogenetics, you need to like, you have your substitution matrix, and if there's biased mutation rates, like if A to T happens at a different rate than T to A, then you have to like account for that.
Right.
And so having a relaxed molecular clock like facilitates incorporating those variable rates without having to have a specific empirically determined rate for each combination. Right, that's what that gives you, and oftentimes you do have a biased rate in one direction or the other, and you also don't know what those rates are. So using the relaxed clock better approximates the accurate rate than using what's called the GTR, the general time reversible model, where the forward and reversible rates are the same. So like assuming that A to T is going to be the same as T to A. That's the idea. So
now that you mentioned, I do recognize.
technical.
Now that you mentioned, I do recognize this image. Cool image.
I use this in class. I've been using this for years.
Integrated synthesis of modern synthesis Darwinism, interesting. Genetics,
Yeah,
mutation.
I think Noble has that in one of his books too.
It's a good figure, and it's a real quick summary of like, here's what we knew in 1870. Here's what we knew in 1950. Here's what we know now. Right?
Wait, okay,
That's
I have
how to
a
interpret this.
I have a quick question. In what context is plasticity used in this
What
plasticity?
was it?
Are you talking about neuroplasticity or is this
Oh
something
no,
else?
that's that right there is phenotypic plasticity. That's
Oh.
like, so you could have two plants with the same genotype, but one of them is in a high shade environment and one of them is in a high sun environment. So the one in the shade environment is going to have an overall like smaller biomass, but the leaves are going to make up a much higher percentage of the biomass compared to the one in a lot of sun. Right, so you can have different phenotypes based on the environment, even if you have the same genotype. That's what we mean by plasticity.
Well, no, well, okay. One thing. Like, I don't know if there's a way to fit that in because, you know what I mean. Just saying plasticity, I don't know. It just seems like it could throw somebody off unless they already know what you're talking about. So you could probably put phenotypic plasticity. So, you know.
Well. So fun thing though, neuroplasticity is an example of phenotypic plasticity more broadly, right? Because you can have two people with the same genotypes in every way with regard to brain development, but their different experiences during life are going to lead to different synaptic connections, right? Neuronal plasticity.
It's not also just that. Like, can somebody mute because I hear something.
Yeah, there's something happening somewhere, but I don't know who it is.
Okay. Let's take twins.
It's me, but I'll mute anyway here.
Well, no, it's not. It's not you. I don't know who it was. Let's just say it was immutable for the fuck of it. But no.
All right, you little fucker.
Hey, I'm just joshing you. But as I was saying, let's take twins for instance. Okay, now they both, they both have the same DNA, but due to different embryonic development, one of them has a higher rate of neuroplasticity than the other. That means one person is more is more able to learn faster than the other. You know what I mean? And learn more. Neuroplasticity is very interesting.
I think that's different from neuroplasticity though, because that's something upstream of neuroplasticity in the process.
Well, but here's the thing. Now, like I'm even less than a layman, okay. But I have looked. I have read up on a few things that interests me. Neuroplasticity being one of them. Right now, and also my experience with people. Like it seems to me that there are various levels of neuroplasticity, right? Because yes, somebody can learn, somebody has the ability to learn, but it just seems like they're only geared to learn one thing and nothing else. You know what I mean? If you say, hey, why don't you study math, and to say, oh no, no, I don't want to study math because of X, Y, and Z, but then
I'm not good at math, right? Yeah.
Well, yeah, they're not good at math, but then there's someone. It's more complicated than making it to be, obviously, but then you have somebody.
What I think you're talking about is upstream of like what we would consider plasticity, though, because the idea with plasticity is you have same genotype, different environment inputs,
right? But
for humans it's hard. But the example that I use for humans is studying. You take two identical twins that got identical grades and always studied together through high school. You stick them in college, and one of them studies for their class and the other one doesn't.
Right?
Who's going to do better, right?
Can these changes be passed on?
I was trying.
No.
Okay. Sorry. Go ahead, Minute.
Okay. As I as I'm trying, as I'm trying to say, right? There are some people who who are born who are born with a higher potentiality to learn. That means their neuroplasticity, that means the neuroplasticity is higher than someone else. You know what I mean?
That's the difference, though. But like having what you're describing is differences in the genetic predispositions to do neuronal plasticity. I'm saying you take two people who have that identically, and they have different study habits. You're going to end up with different phenotypes in terms of memory.
Learning is
whatever they're doing.
Learning is a skill. You learn.
Yes,
you
I
learn
know.
to
I
learn.
know. I know. I know. Learning is a skill. I'm just.
Good to see you.
I'm just saying. There are some people born who are better who are better able to learn than others. You know what I mean? They're better able to pick up information faster than others.
That's not
my
neuronal
point.
Plasticity, though.
If you weren't born with an ample amount of,
but it's not plasticity.
If you weren't born with an ample amount of neuroplasticity, Midnight, you would die within the first 2 weeks. So.
What?
Also, I have a question about that. Neuroplasticity doesn't necessarily have to do with just learning, right? It kind of applies globally. So what?
It's a physical thing.
Yeah.
Can I show you all? Give you all an example that I use in class that I really dig?
But first, I want to say hi to Jefferson. Acknowledge his presence.
So here's the example I use in class for how like learning works. And so this is what I want you to do is I could share the screen if you want, but otherwise I can just do it out loud because it works the same. So picture a blank keyboard, right? Everyone here probably has a keyboard in front of them, cover it up with like a piece of paper or something, or flip it over, cover up your keyboard real fast, right? So everyone's hidden their keyboards. That goes for the audience too. Hide your keyboards. So here's what this exercise I do in class. So what you do is I show a blank keyboard. And in fact, Speed, is it okay if I share the slides? Because it's fun. It's a fun thing.
Yeah, go ahead.
Here you go. I'm going to just give me like 10 seconds. Let me just pull up the slides because I'm going to show it because it's a fun little exercise that I do. And I always get a kick out of the, I haven't done it in a couple of years in person, but I always get a kick out of the in-person response because it's so much fun getting people's responses in person where they kind of flip out a little bit. It's really fun. So give me just 1 second. Okay. So I'm going to share my screen and I'll give so this is learning, right? This is not plasticity. This is learning. This is a different thing. Share the screen. I'm going to share the PowerPoint slide. Okay. All right. It should be available to you.
Okay.
Okay, so we can also, good, we can see my cursor, great. Okay, so the thing here is, can you fill in a keyboard? So what I want you to do, I'm going to show a blank keyboard. What I want everyone to do is start over here with tab, right? You're going to have the tab key, and then just go across and fill in the letters from memory, right? Just one letter at a time, going across, fill in the letters from memory. Okay, that's all I want you to do. Don't move your hands, don't do anything. Just put, fill in the blank keyboard, okay? So we're gonna go. Here you go. Fill in the keys. Everyone looks at this keyboard 1,000 times a day. I'm not going to give you more than like 10 seconds, right? That's how we do this exercise in class because obviously everybody can fill in a keyboard. This is the easiest thing on earth, right? Okay, so everyone's tried it. Great. Stop. How'd everybody do?
Yeah, like nope.
Was everyone able to know, what's this one? What's this letter right here? What's this letter? Everyone here, say it out loud. Turn on your mic. What do you got?
Q. W. E. R. T. Y.
Oh you remember them?
I don't remember.
No one's gonna go. And the gamers will get WASD, right? You'll get that, and then that's it, right?
The only reason
you get QWERTY and WASD, right? And that's it.
Yeah, exactly. Right? That's how I remembered that. Okay. So you're absolutely right.
That's right, exactly. Because so what we just did was, do you stare at a keyboard and try to remember it? No. Okay. Let's try it again. But this time, put out your hands and type the sentence. The quick brown fox jumped over the lazy dog. Who remembered it that time? Everybody know where all the keys are now?
Yeah, I can.
Quick
Yeah.
I can type
fox
it.
jumped over the yeah, everyone knows where all the keys are, right? When you type out the sentence. Because do you ever stare at a keyboard and try to remember it? No.
Yeah.
You never stare at a keyboard and try to remember it. You use a keyboard, and that's how you know it. Anyway, that's what I do. Yeah. Anyway, that's what I do in class to illustrate the.
Something else very interesting is if you look at the keys now, if you look at your actual keyboard and you record a couple of them, okay, and then you go away, you're going to remember those for some period of time. And if you do that several times, or if you go and look at your keyboard and you hold it in your mind for 30 seconds, some part of that keyboard, you will be highly likely to remember it. And now John Maddox has been talking about this Talin paper with the 13. It basically has 13 binary digits in it, and there's a whole lot of work being done on how synapses. Once you fire a synapse, or once you fire a neuron, it is highly likely to fire again, and that happens in your temporal lobe. But there's a whole system there. And the longer you hold it, the more permanent it becomes.
Right.
And
because
anyway, it has
long-term potentiation. Because as you stimulate it, it puts more receptors into the membrane for the neurotransmitters. Yeah, that's all basic phenotypes.
No, it's even more mysterious than that. It happens at a much shorter period of time, and there are no transmembrane modifications at that point. There's only some kind of a modification in the synapse, and basically the presynaptic neuron gets modified, and now they've tracked it down, and it has something to do with actin and calcium and a whole bunch of other things. It's really cool.
I'll tell you what's up. So one, actin filaments are how the neurotransmitters get to the axon terminal. So it's proteins called kinesins, I think, are the ones that basically walk along the actin filaments.
Yeah, they use the microtubules, but
microtubules.
Yeah.
And then the calcium thing is when the calcium, so there's voltage gated calcium channels that open when the action potential reaches the synapse, and it's the calcium influx that causes the vesicles to fuse with the membrane to release
fuse off, right?
Yeah.
Right.
So you put more calcium channels in the membrane, you're going to have more neurotransmitters get released, right? Like that's just.
Yeah, but they're finding that there's some remodeling of the actin cytoskeleton, which has nothing to do with the microtubules. It basically the cytoskeleton on the inside of the synapse, and it looks like they're on to something. But anyway, it's been a big mystery about how this. It's called post-tectonic potentiation, and
what was it?
Post-tectonic
plate tectonics?
No, T-E-T.
Tectonic. Oh, I got it. Yeah, post-tectonic. All right, I'll look that up. Cool.
Yeah, and anyway, so they've been looking for the mechanism for this for a long time because your brain has like all these different levels of memory. It has like a 30-second memory. It has a 1 or 2 minutes memory. It's got a 1 hour memory. It's got a 7-day memory.
Short-term and long-term, yeah.
Yeah, and it's really interesting. And if you fire those things enough times, eventually you're going to make a change in that membrane on that synapse, and that's where you get your neuroplasticity. That's where it actually creates more. I guess on the postsynaptic side, it creates more membrane receptors eventually.
That's that long-term potentiation.
Yeah.
Because you have there's two receptors, and I always get the names backwards. It's NMDA and LMPA, but don't ask me which one is first and which one is second. But you start with one, and then it gets blocked by magnesium, and then the other one comes in after prolonged stimulation. It's
right. And what they were confused about is this is on the postsynaptic neuron.
Right.
That there's this post-tectonic potentiation. So they were trying to find a mechanism for it, and now they're starting to find it, and it's really cool. But Maddox is making a big deal out of it. God did it because...
Matt, can I just say, so I am going to go in a few minutes, but I just want to say that what Maddox does every GD time is he learns something new about biology. That is like, so his like baseline level of knowledge is here, and he learns something new that's like up here, And
Yeah.
then he goes, whoa, this is really complex.
Mind blown.
That had to be designed. That's computer programming. This undermines evolution. I'm like, that's the thing he talks about for like 3 weeks until he learns the next thing that he's never heard of.
Right,
That
right.
everyone in the field learns about in like second year biology in college.
Yeah.
Hmm.
Well,
Right?
it's
Like,
current
that's
thing.
the thing that most non like credentialed yeks do is they like have like these that like the baseline level of knowledge is very low. Like they're not building upwards from a foundation. I'm gonna make this analogy in terms of like building a building, right? You have a foundation, and then you build upwards, right? And you increase your knowledge and understanding as you go. What like Maddox does, and what lots of other YouTube randos do, is you don't bother with the foundation. You just build radio antenna up, right? And they're really tall, but you don't understand anything about it. It's just like, oh, big word. And now I'm going to use this big word for 3 weeks until I move on to the next big word. But there's no foundation. There's no surrounding infrastructure. It's just like one big thing that's complicated that I don't understand. And that's what they talk about for whatever amount of time. And
Yeah,
you
he
can
always
see that
says
in terms
mind
of like
blown.
the cycles. Right, exactly. And you could see this in the cycles of like how they present their things, like, whoa, one big thing that destroys evolution. And it's like, guys, we've been studying this for 3 decades. Welcome to the party, you know?
Yeah.
And it's the same for whatever Maddox does. Every time he reads a new paper about abiogenesis, it's like, wow, did you know how complicated this was? Like, yeah, we do. We've been studying this since the 1980s. Thanks for finally, looking that up.
Yeah, here's a paper that's currently driving him crazy. Now, this guy, he really overstepped his field in that he's making a case for information in the brain that he has not properly made. But inadvertently, they discovered some very interesting stuff. So I'll try to link this in the description, too.
All right, I am going to call it a night. Thank you for having me on to talk about codon bias. I don't get to do that often because I'm not joking.
Coming on, Dan.
I'm not joking. Yeah. Nobody cares about codon bias. So finding people who are asking about it is a rare treat for me. So
I can't.
Thank you.
Okay, what is codon bias before you leave?
Just watch this tomorrow. Just watch this tomorrow.
Yeah.
Okay.
Watch it over. You already did that.
All right. Good night, everybody.
Good night.
Good night.
Okay, I'm putting this paper in the description because this is a pretty interesting paper. And this guy is wrong for the very same reasons that Penrose and Hammerhead are wrong about quantum mechanics and consciousness and sort of thing. But anyway.
You'll see, Speed.
I'll see. Yeah. Well, I had to keep an open mind with this paper because my first gut knee-jerk reaction was to completely reject it. But this guy is doing some decent work. So,
hey, what is he doing? What are you talking about?
Okay. How can I explain this?
Before we get onto that, Speed, I just want to say hi to Brainbug and welcome.
Hi, Brainbug.
Thanks for coming in.
Hey, Brainbug, I just collected I just took like 5,000 bugs out of my freezer and I put the bag out there and I'm collecting another 500 bugs.
We had big mantises on our back door last week. It was pretty cool. They're native too. They're Carolina mantises, so let them back go so they could do their life cycle. But if they were Chinese mantises, I probably would have capped them.
Speed, when you said you were taking bugs out of your fridge, I thought maybe you were just talking about shrimp. But
no, I have those too. But
okay.
No, these are actual bugs.
So if you made a good like shrimp gumbo or something like that, but used bugs instead of shrimp, like wherever it said add shrimp, you added bugs.
There's no rule that says you can't eat insects in the same way that you eat crustaceans. There's really not much difference in them, but we're adverse to terrestrial bugs because we've evolved to react to them with the potential that they're venomous. Just on default, anything with more than 4 legs you want to be apprehensive of because it might sting or bite you, and its venom could kill you.
That being said, I have eaten roasted crickets.
Well, there you go. A man of sophisticated tastes.
They taste like grass.
Really? Wow. So you
I
should
thought
have
they taste like sunflower seeds.
No, sunflower seeds are delicious.
I have a box of roasted crickets right here with me.
Oh, really?
Yeah.
Yeah, we're not doing that.
So how do vegans feel about insects? Where is that within their parameters?
Do you have any vegans in here?
Vegans? Do we have a vegan in the house?
I'm currently wearing a goat head, so I'm definitely not it.
I'm not a vegan.
What's the meme? If we had a vegan in here, they would have told us already.
Oh yeah, there you go. There you go.
Yeah. That's pretty good.
Yeah.
I'm trying to find a good Talin picture.
Rainbug just proved a negative.
But that was we had the whole scientific process to prove it too.
What about vegans?
Yeah.
And none of them being here.
Yeah. Anyway.
I still have a lot of insects and a lot of the world's creatures I would like to eat. Sorry.
You know, one time I bought a book called Dinner with a Cannibal, and it's about cannibalism in the world throughout history and everything, and it's very interesting. But the first chapter is essentially about the things that humans eat in this world that they've gotten used to, and it's amazing.
The aversion to eating insects is cultural. It's not biological. But did this book talk about where the term barbecue comes from?
I think it may, but it's been so long since I've read it. I can't really remember. I've heard that from different sources. One thing they did say was like the monkeys, like if you were watching, have you ever watched Indiana Jones and the Temple of Doom? Monkeys, the scene where they're eating monkey brains in a lot of places, that would be inaccurate because the monkeys are alive at the time in a lot of cultures. And so you're eating their brains while they're looking at you.
This is bad. This is a bad thing. Yeah, I don't know. Let's just not eat bugs, okay?
What's wrong with bugs? They're good for you. They got as much protein as a shrimp or as a crab leg.
Actually, and there are some insects where you get lean protein out of them as opposed to,
mealworms are one of the ones that because their biology has them. They have to burn it really fast, so you get lean protein from.
And you don't get the saturated fat and cholesterol that you get out of crab or shrimp.
And you can stir fry mealworms with.
Wait a minute, no, shrimp doesn't have a whole lot of fat in it, does it?
It has an enormous amount of cholesterol, so it's always been kind of controversial because one of the things that usually comes up is whether or not ingested cholesterol raises serum cholesterol levels. And they know it doesn't. Yeah, but the issue with crab and shrimp is that they have. I mean, if you think that egg yolks have lots of cholesterol in them, you should just take a look at the amounts that shrimp and crab may have in them. And people that regularly eat them don't usually have really high cholesterol levels. So that's one of the indicators that maybe eating cholesterol doesn't change your own serum cholesterol. But there is actually some saturated fat, I think, in both, maybe more in shrimp than crab. But there are other chemical components going on in the tissue that you're ingesting, and so it might be a push.
Yeah, that's the thing about going to alternative lifestyles of eating, which, of course, it's inevitable that we're going to have to consider that given the amount of, methane and land destruction and stuff due to cattle. But where are you going to get your good, healthy fats from to feel sated?
Well, I want to give a disclaimer before we get too much into talking about eating insects, because I don't advocate eating wild-caught insects. Most of them grow under the ground, and when they go through their metamorphosis states, they absorb a lot of heavy metals from the environment. And the higher the pollution levels, the more heavy metals that they absorb. And there's tests on this you can look at. It's insane to see how big of spikes you get with urban populations of like cicadas and stuff. So don't eat wild caught insects.
Yeah, even wild caught fish in a lot of cases, in terms of like the pollutant. Like if it's from the Atlantic, you probably don't want it, unless it's the North Atlantic and it's like a white fish. Even then, it's kind of hit or miss. But like if it's salmon, like if it says, "Oh, it's Atlantic salmon," that's bullshit. You don't want that. It's got a lot of metals in it. You want North Pacific salmon.
Yeah, it's not like you can go back to the good old Neolithic times and just be safe eating bugs or anything out of the ground, because we've pretty much destroyed the ground and the sky and
we've polluted lots of different things, and the problem with that a lot of these companies and a lot of people don't understand that we've polluted an entire cycle of everything. It's a biome that we've screwed up, and that's like you get everything from that, and now it's all poisoned, and you're eating it. So
we can do it just by we don't. Some of these islands we don't even visit, like in like the South Pacific, for instance. We don't even visit these islands; they're isolated, but we have an impact on them because of our pollution in the ocean, and in the air. We've already impacted every inch of this planet, even if we've never set foot there.
Yeah, and to me, that's one of the bigger things. Like, if anything, if people understood that, it's like, look, this is a big push for me. Like, if you want to, it's one thing, people are like, well, I just don't understand what I need to do to save the planet, blah, blah, blah. Like, do you want to be able to eat healthy food? Yes. Okay, then you need to take some environmental steps, because your food comes out of the environment that you're in, so you can't shit in it.
Yeah, it's people celebrating repealing regulations on environmental protection. I just don't understand that. They act like they want a victory. It's the what do they call them? The sovereign land people that are all about that. But you're celebrating being able to pollute our drinking water. It's not, or not, and maybe not even drinking water. Maybe it's just the water we're using to irrigate our crops. Whatever. You're celebrating the ability to put, especially like I said about the insects, heavy metals cycled back out of the waste back into the agricultural water cycle.
It's pretty disturbing.
So instead of sending civilians into space, we should be sending all of our shit and pollution into space, right?
Yeah, I think we need space dumping before we need space tourism.
Yes.
Yep.
Space dumping is good.
Oh, we jettison radioactive waste out into space. I think I've seen that movie before.
Yeah, pretty soon in space, not only will there be bits and pieces of space satellites, there will also be Pepsi cans and Diet Coke cans just floating around everywhere.
I think I think there's a couple reasons why we should do it. One, we don't need that stuff here, right? If we're converting petrochemicals into plastics, and we decide, you know what, instead of leaving them here to plug up the oceans and screw with the wildlife and acidify and all that kind of stuff, we just package them up and we fly them up into space and we dump them out there. Like, okay, it drifts off into space. No big deal.
How much fuel do we burn? Okay, so just what's dumped into the ocean? There's 8 million tons of plastic dumped into the ocean every year.
Yeah.
So the amount of fuel to send that into space, would that be 8 million tons? Would that be?
That's the thing. We'd have to come up with something a lot more
efficient.
Efficient. Something like a space needle or something like that. Space elevator or something like that. Yeah, I think we'd have to have something like that. If we're using petrochemicals to launch petrochemical byproducts in space, we're screwing. We're just losing our minds.
We could use bank teller technology and just put it in those little things and then stick it in there and sucks it up.
I would love that if we had those kind of those negative pressure tubes,
just hoses that stick all the way out
But let's say that we can Superman 4, we can get that stuff out there in the environment. It's out there floating in space and stuff. And then okay, so we got it. It's away from us and it's just floating in the void of space, doing its thing. Maybe it ends up in the sun or something in a couple hundred years or whatever. But let's say that pisses off some alien life forms, right? They show up and they're like, what the hell are you doing dumping all this stuff out here? So here's what we got rid of that stuff. We kept our environment clean and we attracted the neighbors. 2 birds with 1 stone.
Yeah, but what if the neighbors are complaining about us dumping our waste out in the void.
We just explained to them we didn't know any better, and hey, if you guys got any technology to like make this any better, we'd be all about it. But we had no.
Wouldn't it be cool if that's why the aliens come here and kill us because we dumped garbage out there?
Or he's got a point that where they can come here to help us. They can come here and be like, "Here's this technology that converts this back all back into energy. All your hard waste will convert it into energy, and you can burn that."
But then there's a bunch of protests of people saying that that's not freedom, and then they look at each other, they leave, and then they scorch the planet.
Yeah, and printed on their mothership would be a message to us: "Get off my lawn." And it's zap. They kill us.
They just look at us. You had one planet, just one planet.
You fucked it up. Anyway. God, I was just like recycling manically for the last, I don't know, fucking 10 years. And then I started watching like news stories and documentaries about what they're doing with the recycling. And I'm like, it just really, really took the fun out of it for me.
Well,
I mean, they're doing what?
It's a problem around here too, because I live in a college town. So, we have a place where we can take recycling, but since the pandemic, a bunch of stuff got shut down. But then even after that, like there's only certain plastics that they can take. And about 40% of what you take to them, they can actually get out to get recycled. The rest of it just ends up in a landfill. They can't use it. Yeah. So there's a whole network and an infrastructure that's lacking there. And it's kind of sad too, because it seemed like during I don't even remember what decades it was, when it seemed like the push was on for consumers to make the individual choice. Don't throw stuff away, recycle. Do all. If we all do our part, we're all gonna get ahead. And then whatever that upstream chain that was supposed to handle all that stuff after we did our part just collapsed. It's like, well, there's no reason to do that. So now people are right back to just throwing everything into one great big dumpster. And
basically, I just have 2 trash barrels.
Yeah, was that the reduce, reuse, recycle campaign you were talking about there, Jefferson?
Yeah, that was part of it. I think that might have actually been one of the later ones, but yeah.
Like I remember that from when I was in like junior high.
Yeah, me too. Yeah,
it hit a chord with me in that, like, if I got a construction worker and he cuts out a bunch of pipes, like junk fucking pipes, I'll go put them in my garage and I'll save them because I don't want to throw anything away. Because someday I might need that 16-inch piece of rusty fucking pipe, or I might need this screw or this bolt. So I like hoard material, right? Because material is like, I grew up, loving material and working, my dad hoarding material. And so then when it came to recycling, like I have a hell of a time throwing aluminum away. Particularly when they're digging up. Okay, the place I come from, the Boundary Waters Canoe area, that area up there, they're planning on digging that shit up to look for aluminum. And here I am thinking about throwing aluminum away. And it's like, no, wait a minute. Come here and get your aluminum. Leave the fucking Boundary Waters Canoe area alone.
Well, they dig to the landfills and look for the aluminum.
Yeah,
that's what they should do. I mean, the area that you're talking about, are they looking for bauxite or are they looking for like aluminum salts or something?
I think no, actually, it's it's not bauxite. I'm sorry, it's not aluminum. It's copper nickel that they're looking for. So basically, throwing away copper nickel, and they're planning on diverting a river and just doing a whole bunch of shit up there. And the locals are all like, "Give me my gerb. I need my gerb." They're going to have 300 gerbs up there. Gerbs. Which is their name for job, right? They don't know how to pronounce that. So they're all about, give me my gerb, dig that fucking place up. It's like, but the forest they're planning on digging up is just fucking beautiful and pristine and hasn't been touched since the glacier left. Yeah,
they should not. Yeah, they should not do that. Like if you don't if you don't have to take away any forest at this point, don't do it. And there's a lot of places where we should probably be putting forest back, and if they need an example of that, you just point out a time-lapse photo of Madagascar over the last 25 years to illustrate, like, that's a problem.
So you're saying that the glaciers left 3,000 years ago? Speed
5,000.
5,000. Okay.
northern Minnesota, they left about 5,000 years ago. So it was like pure rock up there. That's why when you go up there, if you dig down, the topsoil soil in some places about 10 inches deep.
Does anybody know if we still have any glaciers within the contiguous United States?
Contiguous?
Oh yeah, on some mountains out in Washington or something, aren't there?
Yeah, I was going to say.
Yeah, there's mountainous glaciers, but
They're tiny though, right? I mean, they're not.
Yeah, they're not that huge.
They're not growing back as much each winter.
Yeah.
In general, I'm sure
Did you see another volcano erupted today in some place. I don't know.
In the Canary Islands, La Palma.
Oh, okay. Yeah, did you see that?
Wait,
it's fucking covering a little town. Like,
wait. Did you say the Canary Islands?
Yes.
A little bit of trivia, and for anybody that's listening, the Canary Islands has the world's well, maybe not now, but it did have the world's most pleasant climate. The only thing better used to be San Diego.
Wow.
The average annual temperature in the Canary Islands was 72 degrees. It's sunny. Usually the humidity is below 50%. The dew point's below 50, so it's always comfortable for human habitation. It's considered like one of the perfect places to live in terms of like just ideal temperatures. But given climate change, I don't know what it's been like in the last 5-6 years, but
we wiped out all the indigenous wildlife from the Canary Islands in the Roman era.
Oh yeah.
So what is it?
One of the things the Canary Islands is known for is they have a cliff face or something that, because of volcanic activity, has the highest probability of causing a big shear off the mountain into the ocean and causing a tsunami that will hit the east coast of the U.S. They were they're always monitoring for that with the seismic activity, but this one is not located in a place where that would be a potential problem. Although they do have activity, I think, up north in the area that would.
Well, I hope some states take some precautions, because New Jersey is obviously not prepared for flooding.
Hey, it said here that the authorities began evacuation. You think?
Yeah. It's going at what they about. Okay, I'll do the conversion. It's going to about 0.62 miles every hour, apparently. So it's a slow mover, but they need to still get out of the way ASAP.
And the volcano in Iceland's been doing some pretty cool stuff lately too, and it just went dormant again.
Yeah, that one. There's a YouTuber named Wild Eden, and she keeps telling me about this. She keeps watching the live feed thing.
Oh, I love it. Yeah, Gutentag runs a thing every day, every few hours on it. He does drones, and that thing they had to evacuate a bunch of people. It went dormant for like 4 or 5 days, and then it blew up. It erupted all over the place, not out of the caldera, but like further down the lines. And they had something they call hornitos, which are like lava tornadoes that came up, and then it got vicious, and then it just went dormant a few hours ago, about 12 hours ago again. It's like, I watch it every day because it puts it watching volcanoes erupt slowly puts you to sleep. I mean, it's very relaxing.
Well,
very relaxing.
It was one of the creepiest things that I'd ever heard. I can't remember what book I was reading that had that little tidbit in it, but the caldera that's under Yellowstone in the United States.
Yeah.
If it erupts like a volcano, it's a planet killer.
Yeah.
Yeah. At least United States killer.
Yeah, the biome is gone. Maybe 2% of the world's human population makes it. Maybe 2%, because of the nuclear winter type effects that it issues in after it kills everybody. It kills off 3/4 of the entire United States. Like from where I am back to Maine, we might live for 3-4 months.
Yeah,
after it erupts.
And it erupted it erupts like every 700,000 years roughly, and I think last time it erupted was 600,000 years ago or something.
Mm-hmm.
And it's like, yeah, they have scars going all across Washington as that hot spot is moving into where the Yellowstone is now.
correction, the hot spots in the same place. It's the continent.
Oh yeah, it's a continent. Okay, Nick Zentner.
All right,
You've been watching Nick Zentner, haven't you?
I just need to apply my learning.
You're learnedness. So David Neff is having a stream now. Everybody should go watch that, or not a stream, but a thing.
Brainbug,
What is
is that your supper?
Oh, supper.
Are you hungry?
I am hungry. That looks pretty good. That looks tasty.
Looks like a lot of fiber.
Yeah. Well, there's some protein mixed in here too. These little guys. Oh, come here, you. He's all grumpy.
What have you got going on there?
Ooh, I see something. What is that?
Scorpion.
What? Don't touch a scorpion on my.
Scorpions all the time. I got more scorpions up under here. See?
Do they bite you? Or sting you?
I try to get them not to bite me.
Well,
most scorpions don't bite, but they can pinch and they sting.
These ones would probably sting.
So, Brainbug, was that your favorite rock group from the 80s? The Scorpions?
Scorpions? Yeah, they rocked me like a hurricane.
Yeah, pretty good.
Yeah. Did you ever see them live?
I never did. Never had that opportunity. They
They
were on MTV a lot.
That was a little bit before my time. I was still kind of a kid when they were hitting it big.
Yeah, I was like, a little bitty. I'm a couple years younger, so.
Yeah.
Yeah.
Now, Def Leppard.
Shit. Oh, shit. Hang on a second.
What's the matter?
It's okay, guys. He probably just clicked on the wrong thing. He's got browser Tourette's.
Y'all see my rolling tray over there? Good grief.
That's a pretty energetic scorpion that you got in there.
Yeah, this one's sleepy. He doesn't even want to wake up, but I'm gonna leave him alone. These two are playing.
Don't wake a sleeping scorpion.
That should be a real saying, shouldn't it?
Well, we lost Dessel. Don't we need to warn him about all the scorpions out there in New Mexico?
I don't think that he wouldn't know about those.
So I grew up with movies on TV where they were killing people with scorpions, so I thought they were like, deadly, lethal.
That's what ninjas would use to, bump you off silently.
Yeah.
Shit. I mean, that's why Scorpion from Mortal Kombat is a thing. That's a late 80s, early 90s kind of thing,
Oh my gosh, when I was a kid and I wanted to pet scorpions so bad. I was like, oh, that'd be so cool.
You wanted to pet him?
I wanted to pet a scorpion.
There was something wrong with you.
Isn't it kind of weird though? Like most of the stuff that we thought was dangerous, like because I'm pretty sure, like as far as predators go, it's like there's a North African cat, and it looks like an adorable kitty, and it's like based on its weight and the amount of time it spends hunting, it is the world's most efficient predator.
Wow.
And like nobody has that thing on the back of a jacket. We've got a scorpion, we've got rattlesnakes, cobras, great white sharks, all that kind of stuff, because they can do a lot of damage when they actually do something. But half the time they don't really do anything. And this cat goes out every night, all night, and murders the fuck out of its environment. And it's adorable.
Yeah, that's kind of like Loki here. I'm petting him right now. He goes out and murders every morning.
It's the adorable killers you got to look out for.
Cats are invasive species now.
They are.
Mm-hmm.
Well, he doesn't always murder. Sometimes he just brings them to me and lets them fly away or jump around or whatever.
Well, they did that with a chipmunk, right?
Yeah, the chipmunk and a couple of birds this week that I had to capture and release. We had a bird living in the house for 3 days and we couldn't find it. We could hear it, and I finally got it in a net and got it outside. But
the worst is when they leave something semi-living in front of you.
Oh yeah.
And then they yell at you, like they meow at you, and you can't figure out, like, do you want help or do you want me to finish it off? Like, it's like, prove to me you can kill, like,
yeah.
Yeah, well, they come in and they announce, you know, meow, meow, meow. I got something in my mouth, right? Well, when he came in with that chipmunk, he was like, meow. Like he wasn't sure. He wasn't really sure he wanted that fucking thing in his mouth.
Identify
right?
and neutralize.
And then he let it go, and then it took all day to capture it. I captured it during a live stream. Just, I don't know. Anyway, so I was gonna show this volcano erupting. Let's see if I can do that. Full screen. I think full screen. I think that might work. Yeah, that might work. I think you can see it. Hopefully they were showing me a diaper ad. Like, wow. Wow, that's kind of cool. I mean, you'd hate to see that in your backyard. And here's people with their phones, you know. I mean, it just blew out of it, you know, and like it started burning some of these houses. Yeah. I suppose I'll get a copyright strike for this. Holy shit! Look at this. That's some lava.
It's amazing.
Yes, it is. Okay, so enough of that. CNN, eh? I'm sure I'll get a copyright strike for that because I didn't play the diaper ad. If I'd played the diaper ad for you, what the fuck are they advertising diapers to me for? I'm 70 years old. I don't know. Oh, I know, because I'm 70 years old.
That's because I'm on here and I frequently tell people that, you know, Dark Dawkins' diaper is full.
Yeah.
That could be it.
Yep. So I think David Neff is still going on over there. I think we should shoot her in the head. What do you think, guys?
Yeah, that's a good idea.
You need to play that. I just put a link in private chat. You need to play that video with the chipmunk.
Okay.
Play the first minute of it. Watch the chipmunk. So this is what this person did to their chipmunk.
Okay. Let's see. Share. Okay, let me first of all do this. Share screen. I'm working on it there, David. Ooh, it's like all Chinese. Okay, ready?
Yeah, watch what happens. He opens the door. This is what he did with his chipmunk. Here's his chipmunk, comes in the house.
Little fuckers can run. Oh, nice.
He's got nice. And I think I didn't watch it. I saw this right after when that whole chipmunk thing happened. I saw that. I haven't watched the whole thing. It's like an 8-minute video.
Okay.
We're got a little pet chipmunk. I was gonna give it to you the other day, but yeah. But I was like, oh, this is what you should do. And the title of it is, I don't know, I built a house for a chipmunk or some fucking thing. When I saw that, I'm like, oh my God, I got to give this to Speed.
Just so you can keep it from shitting in the house.
Yeah.
That's pretty cool.
Funny. Yeah. And then, he was just about to go out the door. He was just about to let him out right there.
Yeah. By the way,
as far as I watched.
I got to mention that video, the text that was going, that's Korea.
Oh, okay.
Yep.
I wouldn't know the difference because I'm an idiot.
That's it. Not a lot of people do. I just wanted to say that's South Korea.
Okay, boys and girls.
It's good to have you in with us here, Jefferson. You can
you straighten us up.
Bring all that wide knowledge and experience that you have.
I'm a worldly bastard and a complete white trash hillbilly. Yep.
Okay. We're going to shoot it in the head. Goodbye, cruel world.
Thanks, everybody, for popping in.
Good night, and